Hello, everyone. Good afternoon on the East Coast. Let's see how many colleagues we're having all joining. The number is growing. So give 2 more minutes and then we can start. We want to appreciate your participation here today in this presentation. Thank you so much for taking the time. We had close to 200 registration. So this is the number that we are expecting today. But of course, the link for this recording is going to be available as well for us to share with our clinicians and colleagues. So I hope you enjoy. We are around 100 guests right now. So I think that it's yes, it's about time for us to start. This way we can be mindful to our time and not delay with the presentation. So let's get started, team. You're okay? Okay, let's do that. Today we are here to discuss a new platform of PGTA. So the name of this platform is Smart PGTA Plus. And our intention this afternoon in the East Coast is that you can understand the applicability of this new platform and how this can help us with the better clinical results in our clinics. So Alyssa, if you can move to the second slide, please, and then I can introduce our team here today. So myself, my name is Juliana Kutsi. I'm the moderator here today. My position with iGenomics is VP of Sales and Marketing. I oversee North America. It means Canada and USA in our territory division. I'm a PhD geneticist, but I have been working with business development for a long time already. And I have been around and in the market of pre-implantation genetic testing for more than 20 years. So I hope that we can share interesting information with you today. So moving to our presenters, I have here the two Alyssa that I'm very proud of and then I'm very happy with. So starting with Alyssa Snyder. Alyssa Snyder is also a PhD geneticist with a background on genetic counseling. She has counseled many patients already on PGTA. And today, her position is Director of Clinical Genetics Service with iGenomics. She has been with iGenomics for more than 10 years with a vast experience in the field. Also, we have with us Alyssa Magwood. Alyssa Magwood is a scientific advisor and business development with iGenomics. Alyssa has a background in molecular biology. She has been working on research and molecular biology for many years in the private or even public domains, coming from universities. And Alyssa Magwood is based in Canada. Alyssa Snyder is based in the West Coast in West California. In the end, on our discussion in our panel for questions, we have the presence of our colleague Shannon. Shannon, thank you for being here with us today. Shannon is an embryologist. It was like a clinical embryologist for more than 25 years before she joined Vitrolife. And she has extensive knowledge in the process and the protocols inside the clinics as an embryologist. She works with iGenomics and Vitrolife on technical support and education of our customers. And her title is Clinical and Scientific Support Specialist at Vitrolife. Thank you, Shannon, for coming. So next slide, please. And let's get started what we came today to discuss. After our experience with the first ASRM together under the umbrella Vitrolife Group, I thought that it would be interesting for us to discuss a little bit the two brands in 2 minutes slides. Alyssa, the next one. I think I can, yeah. So Vitrolife Group, it's now the bigger organization that keeps the two brands, Vitrolife and iGenomics. So the two brands coexist under the Vitrolife umbrella. And we have the same objectives and the same vision and mission. And what we really want is to fulfill the dream of having a healthy baby. If you're curious to know more about the Vitrolife Group, you know that we are a public traded company. We have around 1,100 employees, and we are a global company, and we are assisting more than 100 markets. Vitrolife was founded in 1994. Igenomix was founded in 2011. So the two brands are working together and collaborating to better serve our customers and to bring solutions to the market. When we talk about Igenomix and the PGT laboratories, that is the subject of our presentation today. In North America, we are present with the 4 laboratories. We do have our Canadian laboratory based in Toronto. We have a New Jersey laboratory, Miami, and Los Angeles. So this is all the accreditations. You can see that several of them, they are redundant because we do have a plan of redundancy and contingency plan for difference that we have in laboratories, for necessity to moving samples or to overcome any type of trouble that we may have on the technical aspects. Besides that, Igenomix has several other laboratories outside of US and Canada that can give us support as well, and they have licenses as CLIA, as CAP, that it's helping us as well if we need to export any type of biological sample. So moving to Smart PGT-A Plus, what is that? I'm not spending time on this slide. Every time that we see a slide that starts with the fish, everybody was thinking, oh, here she comes with all the explanation of the story of the PGT-A. It's nothing like that. I just want to jump to our NGS, but I need to recognize, and we need to recognize, that the field has been evolving a lot. Even with the discussions of mosaicism, not mosaicism, should be reporting, not reporting, what is the accuracy of detection for segmental, chaotic, complex abnormality, and the discussions, they go from technology to clinical application of the test. But what it's important for us to understand is that it's a field ever in evolving, and with the technology and the AI and the algorithm, more data we are adding to those analysis and to those algorithms, we are getting to a better result or even a more accuracy result in when we are reporting an embryo and what we want. What is the embryo that we should be transferring? What is the embryo with more likely to implant? What is the embryo with a higher chance to result in a healthy baby? So today we are talking to you about the analysis that Igenomix is offering, combining the platforms of NGS and SNP analysis. Our intention with this platform is bringing 3 new information that our regular PGT-A was not being able to report to us so far. So in the next slide, we are going to see that our regular PGT-A, Igenomix has been naming our regular PGT-A or conventional PGT-A as a smart PGT-A for a long time. Smart comes from our algorithm. Since we validated our technology and we published the validation of our technology in 2020, we have been naming this test as a smart based on the capability that we have to be consistent across the globe with all our laboratories. Our limitations and thresholds are exactly the same, whatever we are, because our reporting system, it's very automatic and integrated with our algorithm and database. So our smart PGT-A was always able to report aneuploidies, mosaicism, single aneuploidies, chaotic mosaics, and segmentals. What we are adding today is the capability to detect ploidy, what we are naming here as the genetic PN check. So the capability to detect triploidy or haploidy. We are also adding the capability to detect contamination with exogenous DNA. We are able to see if the DNA that we have inside the tube are DNA coming exclusively from the embryo that was biopsied, or if inside the tube we have contamination with any other source of DNA that can be a confounder of our results. And also we are able to compare the embryos that we are receiving inside a cohort and detect if those embryos are siblings. So the combination of sibling QC and contamination is bringing to us a much more reliable test. In comparison with the regular PGTA and the PGTA Plus, we can see here the features that we named add-on on the table as the ones that we are bringing as new under Plus, that are the features that we depend on the SNP analysis to be able to detect and to report to you. So again, they are ploidy, presence of contamination, and siblings QC. What we hope to offer with this new platform, it's a technology again that it's totally based on AI and totally based on our algorithm for reporting. You should not expect difference or discrepancies on your reporting, because it should be a very consistent and stable technology across the laboratories. This is what allows us to use any of the laboratories that we have for the analysis, because the system is the same. With the inclusion of the SNP analysis, we hope to have more enhanced accuracy and more confident results, because the number of the analysis, the number of data that we are using for our analysis is higher. We maximize the likelihood to have a successful pregnancy, because we are separate triploids and haploids from our diploids embryos. We reduce the risk of miscarriage for the same reason. We are definitely enhancing the accuracy of our diagnostics. We are ensuring a better quality for the laboratories. Contaminations and siblings can be used as KPI for the clinics, can be used as KPI for us to give us the assurance that we need that the reporting is coming from the embryo DNA, not other source of DNA inside the tubes. And we provide the confirmation that all embryos in that cohort are genetically related. It shows, or it seems that I presented everything that we have in the new test already with the 2 slides, but definitely want to understand how it works. We want to understand more details about the reports and what it's the clinical implications for everything that I just said. So with that, I'm transferring the floor to Alyssa Snyder, that it's going to start with us with the clinical implications. I hope you like it. In the end of our session, we have a time for questions. Please don't be shy. And if you want to start typing the questions right now, I'm taking care of it. And I'm going to just close my camera and give the floor to Alyssa Snyder.
Thanks so much for that overview, Juliana, of Vitrolife and iGenomX and this new test that we're all so excited to present to our clients today. So as Juliana mentioned, I'm going to be presenting to you a little bit more about the clinical implications of the ploidy analysis and the genetic pronuclear check. So as everyone knows, PGTA is essentially an embryo selection tool whereby the chromosomes are assessed to determine which embryos have the highest reproductive potential based off of having the typical number of chromosomes, and which embryos are not good candidates for transfer because they have a chromosome abnormality, an aneuploidy, and extra missing chromosomes that renders them unlikely to develop normally into a healthy pregnancy. So first, an egg and a sperm, both of which should have 23 chromosomes and be haploid, are combined in the dish to create, to fertilize an embryo that is ideally having 46 chromosomes, or a diploid embryo. Next generation sequencing, or NGS, looks for relative copy number variations. The average number of reads is normalized to copy number 2. For aneuploid embryos with extra missing chromosomes, we see elevations or depressions in the number of chromosomes, in the number of reads for that particular chromosome compared to the rest. And it's really important to note that what we aren't able to detect is a change in, we're not able to look at the chromosomes themselves. We're not looking at a picture of the chromosomes, and so we're unable to detect complete changes in chromosome number if it's across the board. So the basic limitations of PGTA testing that we are addressing with PGTA Plus today are these 2. Ploidy abnormalities, because the DNA is normalized, a change in the number of chromosome sets cannot be observed. Again, NGS is looking for relative changes between the chromosomes. So if you have an increase of the total number of chromosomes or a decrease in the total number of chromosomes, NGS is unable to detect that. And another limitation of PGTA is that the accuracy depends on this assumption that the sample represents the tested embryo. And there are a few reasons that this assumption might not be true, the most common reason being a biological phenomenon called mosaicism. Other reasons are procedural, including contamination and sample mix-ups, where PGTA Plus has the ability to help us. So PGTA Plus is an advancement in technology aimed at addressing these two limitations to further increase the accuracy and the confidence of the results. I'm going to be talking with you about ploidy abnormalities in my next few slides, and then I'll be handing the slide decks over to the other Alyssa on the call, who will be talking a little bit more about how we can address contamination and sample mix-ups. The new technology harnesses the power of NGS and incorporates an additional step to enrich for SNPs. So SNPs, or single nucleotide polymorphisms, are changes in a single nucleotide. They are distributed across the genome, and they're frequently polymorphic between individuals, allowing for a genetic fingerprint to be analyzed. The vast majority of SNPs have only two alleles, so for example, either A or T, C or G, and we call the major allele A and the minor allele B. In diploid embryos, the expected allele frequencies are 0, 50, and 100. So you could have AA and the allele frequency for B would be 0, or you could have AB and the allele frequency would be 50. For triploid embryos, on the other hand, the expected allele frequencies are 0, 33, 66, and 100. PGTA Plus uses custom NGS panel targeting 357 different SNPs. And by looking at the distribution of these allele frequencies for the SNPs in an embryo, the algorithm can diagnose embryos as either diploid or triploid. Alyssa is going to be showing you these profiles again when she's talking about contamination analysis. So check it out. It's important to note that these allele frequencies are 50 and 100 and 33 and 66 for the triploid embryos. So a normal ploidy is diploid with 46 chromosomes in total. You've got 2 sets of every chromosome. A haploid embryo has only 1 set of chromosomes, one chromosome of each. A triploid embryo has 3 sets of chromosomes, 69 chromosomes in total. Haploidy and triploidy are actually compatible with pre-implantation embryonic development. So these embryos can develop into a blastocyst that could be biopsied. But they are not compatible with normal development to term. And triploidy can be found in 10% of chromosomally abnormal spontaneous miscarriages. This is what an embryologist sees. So when we think about diploidy, we expect 1 set of chromosomes coming from the egg and the other set of chromosomes coming from the sperm. And those pronuclei come together at the fertilization, and we can actually see this on the pronuclear check. Sometimes we don't see these 2 pronuclei. Sometimes we see 0 pronuclei. Sometimes we see only 1. Sometimes we see 3 distinct pronuclei, and sometimes we see 2 plus this additional circle, which could be an additional pronucleus or could be just granularity in the embryo. So embryos that have 2 PNs are what we would consider to be normally fertilized. And 0 PNs, 1 PNs, 2.1s, and 3 PNs are what we would call abnormally fertilized oocytes. And our comfort level decreases as we go with these different pronuclear statuses. 2 PN embryos, we have high confidence and comfort that these are probably diploid embryos, but the rest of these we don't know if they're diploid, could be triploid, and so usually these embryos are not used for clinical use. Unfortunately, even with normally fertilized embryos, there can be undetected ploidy abnormalities. So these 2 PN embryos can develop to the blastocyst stage, can receive a euploid PGTA report, especially if it's 46XX. There's no relative copy number variations detected. And these embryos can be transferred and then result in implantation failure or miscarriage, molar pregnancies due to undetected ploidy abnormalities. The incidence of triploidy in normally fertilized embryos is thought to be low. But iGenomics set out to quantify the incidence of ploidy abnormalities among normally fertilized euploid embryos that would otherwise be candidates for transfer. So in this particular study, a total of 1,075 euploid biopsies were assessed for ploidy at 2 different laboratories. The incidence of triploidy was found to be 0.79% in Lab A and 2.5% in Lab B, for a total incidence of triploidy detected at 1.86% for normally fertilized euploid embryos. No haploidy was detected in these embryos. So these could be embryos that would have been transferred without the additional analysis of ploidy. On the other hand, after a pronuclear check, embryos originating from abnormally fertilized oocytes, for example, those with 1 PNs or 3 PNs, are usually discarded and not considered as candidates for transfer. And these could potentially be embryos that are lost from the ability to use for clinical use. So we set out to quantify the frequency of diploidy among abnormally fertilized embryos. So in this study, abnormally fertilized oocytes were developed to the blastocyst stage. 318 biopsies were submitted for NGS analysis. 58% were found to be aneuploid, and 41.8% were found to be euploid. And these 133 embryos then underwent ploidy analysis. The majority of 0 PN derived blastocysts are diploid and can be good candidates for transfer. And this supports the general confidence that embryologists have with 0 PNs. So we can feel good about 0 PN embryos. But we do see some haploid and some triploid embryos in this data set. On the other hand, 1 PN embryos and more than 2 PN, so either 3 PN or the 2.1 PN embryos, can also be diploid. About a third are found to be diploid among these abnormally fertilized oocytes. So here we have 35.6% of 1 PN derived embryos are diploid. As expected, the majority are haploid. On the other hand, these 2.1s or 3 PNs, the majority are diploid, but a solid 33.3% were found to be diploid. And according to this data set, the additional analysis of ploidy detection allowed for the clinical use of 72 additional euploid diploid embryos that were otherwise not considered for transfer. So the purpose and the allure of ploidy analysis and this genetic PN check is to simultaneously increase the number of diploid candidates for transfer after abnormal fertilization or no fertilization, and simultaneously decrease the number of embryos that are transferred that have undetected ploidy abnormalities. So any couple could benefit from using SMART PGTA+, but the following indications are especially relevant. So we want to be able to rescue embryos that have abnormal fertilization that could actually be diploid. We find that ploidy analysis is especially helpful for patients with a reproductive history of haploidy or triploidy, patients with a complete or partial molar pregnancy. So those partial molar pregnancies can be triploid and complete molar pregnancies can be diploid. And if the diploidy arises from uniparental disomy or one sperm endoreduplicating, this will actually look like a haploid result, and so we would be able to detect that as abnormal. And then also patients with severe male factor or a high rate of diploidy in sperm can increase the chance of ploidy abnormalities in the embryos, which we can detect. And so with that, I'm going to hand the floor over to Alyssa Magwood, who's going to present on embryo QC. And I'll stop sharing my slides so that she can share hers.
Okay, thank you, Alyssa. Let's okay, while we're waiting for the slides to come up, and Alyssa, if they don't pop up, maybe I'll just get you to start sharing again and then we can walk through it together. So just to sort of reiterate where we're at now in our discussion. So as Juliana mentioned at the beginning, SMART PGTA Plus is made up of those 4 components: the SMART PGTA, and then 3 new features ploidy or PN check, which Alyssa Snyder just went through, and the last two, the contamination piece and the sibling relatedness piece, which make up embryo QC, and that is what I'm going to share with you right now. So let's see here. Alyssa Snyder, if you just can confirm that you can see my slides.
You should share your other monitor if you can, because I can see the notes version.
Thank you. How about now?
There you go. Yep, that looks good.
Perfect. Okay, so let's tackle this interesting piece of embryo QC, because I think it's very intuitive from the information Alyssa Snyder just shared with us, how important the ploidy analysis and the PN check can be. Now, my role is to show you about the importance of the other two pieces. And so I think that one of the easiest ways to think about this piece of the discussion are to reflect on what are some reasons that PGTA can be less accurate. And Alyssa and Juliana sort of alluded to these earlier on, but let's go through them now. So first and foremost, does the trophectoderm biopsy that an embryologist takes, does it actually represent the inner cell mass, the developing baby? Of course, if it doesn't, then that makes the test less accurate. However, we know this is a biological question and with a biological answer, and something that at this present time we can't address. But what about the fact that the contents of the biopsy tube may contain DNA that comes from other sources other than the embryos? Or another way to say that, do the contents of the tube contain embryo cells only? And so some other possible sources of DNA, of course, can be the maternal or the cumulus cells that might persist despite your best efforts to denude the egg and wash those cells away. Also, we have to talk about DNA or cells coming from anyone who handles that biopsy tube. So that would include embryologists, or, you know, on the testing lab side, the technicians that are receiving those samples and performing that PGTA. And of course, that is a procedural possibility. And then finally, we don't like to talk about this, but what about sample mix-ups, mislabeling of samples, or tube switches? And of course, this is also a procedural effect. I just want to take a minute to emphasize and to speak about the fact that this concept of detecting these things that make or can make PGTA less accurate. So the presence of external DNA, or this question of possible sample mix-ups, these are not entirely new concepts. I think we all have a sense that these things can happen. And in fact, on the genetic testing lab side, we sometimes see evidence of these inaccuracies or of these potential problems. iGenomX is a pioneer in non-invasive PGT testing. Our test is called Embrace. We've been working on this test for close to 7 years now, and through our experience with Embrace, with non-invasive embryo testing, we've really pioneered the detection and identification of contamination, because as you can imagine, an embryo releasing cell-free DNA into a small media droplet is very prone to contamination from, of course, maternal cells, but also from anyone who handles and or is around that droplet of media. And so we learned a lot through non-invasive embryo testing, through Embrace, and we learned a lot about the sources of this potential contamination, and of course the fact that it does exist and how we can identify it. Secondly, PGTA discrepancy cases. iGenomX has a wonderful protocol to handle any PGTA discrepancies. And by discrepancies, I mean, imagine that your patient successfully becomes pregnant. Maybe they do a non-invasive prenatal test, an NIPT, and they find out that the sex of the baby is male. However, their PGTA result suggested that it was female. Or perhaps they had a euploid result and they unfortunately suffer a loss, and they test the products of conception and find a discrepancy there. These things are very rare. It's important to emphasize this. Very rare, these discrepancy cases. However, in our experience with discrepancies, we have found contamination of the PGTA sample, and we've also found sample swaps to be the cause. And so we have experience with this. And lastly, this concept of intermediate XXY profiles on NGS. And what I mean from that is, remember or think back to the NGS profiles you've seen before, and a few minutes ago, Alyssa Snyder shared some with us, where we normalize all the reads to the 2-copy line. And if there are losses of chromosomes or gains, we can see that. What we mean by intermediate XXY are when instead of 2 X chromosomes for a female or an X and a Y, a single copy of an X and a single copy of a Y for a male, we see these intermediate copy number changes where the reads are not aligning to where they should be. And one of the possible reasons for intermediate XXY profiles is, again, contamination. And so we have methodologies that we can use to identify and determine what the cause of these intermediate XXY profiles are. But one of the causes is contamination of the sample. And so taking all of the experience that we've had at iGenomics with, identifying and determining these possible problems with PGTA, we're reflecting back now on the development of the PGTA Plus test and asking ourselves, okay, so we understand that there are potential inaccuracies. What can we do about this? How can we improve the test overall? And so, of course, this question of does the trophectoderm biopsy represent the inner cell mass? At this time, there's really nothing that we can do about that. However, we can address the possible other 2 things that can cause PGTA inaccuracy. The first, of course, if we want to determine does that biopsy tube contain embryo cells only? Well, we can add on an analysis that helps us to detect the presence of external DNA, or essentially looking for the presence of 2 genomes instead of 1, as we would expect if it's embryo cells only. And then as far as addressing the sample mix-ups, we can add on an additional analysis to help us confirm that all the samples from one patient are genetically related to each other. And this analysis will help us in addressing the possible issue of sample mix-ups. And so, of course, you've seen the slide already when Juliana showed it, but just to emphasize again, when I speak about embryo QC, I'm really talking about these 3rd and 4th components of the SMART PGTA Plus test, the contamination piece and the sibling QC piece. And so let's start with a little bit deeper dive on this topic of contamination, or identifying the presence of external DNA in that tube or 2 genomes. And so like any test that iGenomics develops or brings to market, we have extensively validated this contamination detection. And so just for some information, and please, of course, I need to say that if you have deeper questions or you would like more information on the validation of the contamination or the validation of sibling relatedness, which I'm going to share in a minute, or anything that we've talked about today, please contact your scientific advisor, and one of the scientific advisor team members would be more than happy to review this data with you in depth. But for the purpose of this presentation, I'm just going to quickly skim over the validation so you get the general idea. So we selected 84 parameters to build this ploidy contamination model, and we used a total of 2,500 different samples to develop it. So on the lab side, we actually made mixes of 2 different sources of DNA, of 2 genomes, and we made those mixes anywhere from 20 to 80%, and then we analyzed that contaminated DNA. And then on the data analysis side, we made in silico mixes of BAM files, and we were able to detect contamination in these BAM files from contamination ranging from 10 to 90%. And so the test has a sensitivity of 100% and a specificity of 98.8%. Just to remind you of what Alyssa touched on earlier when she was discussing the ploidy analysis and this variant allele fraction distribution. You'll remember she told us that a diploid sample will peak at around 50% here, where a triploid sample will show us peaks at 33 and 66. And if you look over here at this series of variant allele fraction distributions, you'll get a good sense of what we see with different levels of contamination. And I will just draw your attention specifically to this graph of 50% contamination. And what you can see here are peaks at 25% and 75%. And those are real flags for us. And of course, you can see all of the different percentages of contamination listed here. But so this just gives you a general idea of how we have validated the contamination detection piece of the test. And so the good news, After our validation and then some clinical samples and things we've been working on, we know, or we've seen, that an estimated average of 99.2% of biopsies are free of contamination. And this is great news. I think we know, and we pride ourselves on the fact that our IVF labs and our embryologists are extremely careful. Of course, on the testing lab side, we take every precaution we can, and we are also extremely careful. And this is reflected here in this very good news that most biopsies are free of contamination. But on the flip side, contamination is expected to be found in about 0.8% of samples, or if you do the math on that, it's about 8 out of 1,000. And if you are testing, as iGenomX does, tens of thousands of biopsies every year, you can see that this number of contaminated samples and potentially affected patient results can really grow. And so it is an important thing to address. And so when we talk about contamination, we have sort of two different scenarios that we can encounter. So imagine this first example where we have what we call a DNA admixture. And basically what we have here is a sample or cells from the embryo, so some embryonic DNA and a contaminating amount of exogenous DNA. So maybe it's cumulus cells or maternal cells, maybe it's embryologist DNA, maybe it's an iGenomX technician DNA, hopefully not to any of those things, but there's a mixture of a DNA admixture. And so of course you can imagine that depending on what that mixture is. So imagine that, and we assume that any exogenous DNA contamination is going to be euploid. The maternal genome is most likely, of course, euploid. We are all presumably euploid as well. And so any contaminating DNA is most likely to be euploid. So depending on how much contaminating exogenous DNA is, you can imagine what the impact would be on the result for the embryo DNA. And so that's why we say that this could lead to potential misdiagnosis, because the NGS result will be an average of these two sources. And depending on how much embryo DNA you have and how much contaminating DNA you have, this will be the result that PGTA generates. Now, our new SMART PGTA Plus test, which adds on the SNP analysis, will actually be able to detect and call out the presence of two different genomes. And we can do that when we see anywhere from between 10 and 90% contamination in a DNA admixture. Now imagine the second scenario that we can run into, and that's pure contamination. That's when the sample actually doesn't contain any embryonic DNA at all. And so it would be 100% external DNA coming from, again, the sources that we've talked about before. And of course, the risk with this situation is complete misdiagnosis, because the NGS result will not represent the embryo at all. It will represent the full component of the contamination. And so, of course, our PGTA plus SNP analysis won't be able to detect this, because it will detect only a single genome. However, if that contamination is not of maternal origin, the SNP analysis might catch this as a sibling mismatch. And so now I'll take a moment to talk to you a little bit more about sibling mismatch and what that can look like. So I've used the word sibling mismatch. We sometimes call it sibling QC, sibling relatedness, all referring to the same thing, determining if the embryos from one patient have a genetic relatedness to each other. And so this essentially is embryo fingerprinting without the need of samples, additional samples from the egg and the sperm provider. So this test does not disrupt the flow in your clinic at all. We don't ask for any additional samples. We're doing this fingerprinting without DNA from the egg and sperm provider. And again, it assesses the genetic relatedness between the embryos that are in the same cohort. And it can confirm that all the samples analyzed originated and from the embryos that were fertilized by the same sperm and the same egg source. And of course, if you remember back to Alyssa's conversation about variant allele fraction distribution, we need to have at least 2 diploid embryos in this cohort to be able to properly analyze sibling relatedness. Just like our embryo QC piece, we did extensive and thorough validation on this prior to rolling it into our SMART PGTA Plus test. And again, just briefly, we analyzed 613 diploid samples, and we used almost 5,000 sample pairs for every single run in over 40 NGS runs to develop this model. And we used ethnicities, a varied amount of patients from different ethnicities. So in this example here, you can see we've got 5 embryos from 3 different families. And after we run them through our sibling relatedness model, it correctly assigns each of the 5 embryos into the correct family that it's associated with. And so again, let's start with the very good news here. After our validation and some, working with clinical samples, we see an estimated average of 99.8% of trophectoderm biopsies are correctly identified. And again, I think this won't come as a surprise to any of us, but it's also very reassuring to see that we are extremely careful on the IVF lab side and on the iGenomics testing lab side. We are extremely careful, and we have our own QC checks in place to help us avoid sample mix-ups, but they can still happen. So we see apparent mismatches in about 0.2% of samples. So again, that would be about 2 in 1,000. And what are some potential causes of these apparent mismatches? And we've just listed a couple here for you to think about. Embryos derived from different donors is a possibility. True mismatches where the incorrect embryo was biopsied or tubed, perhaps the tubes were mislabeled, or potentially the tubes were swapped. And the last possible reason, which we expect to be more rare, and again, this is something you could reach out and discuss with your scientific advisor, but it would be the biological variation in SNPs. And this can especially be a problem when we are only looking at 2 embryos. And so this test really draws its power from the more embryos in a cohort. So the more embryos that you have in a cohort, the more, the better the algorithm is at detecting the sibling relatedness. Okay, so what are some benefits of the embryo QC? First, of course, I hope that I've convinced you that embryo QC really plays a role in the accuracy of PGTA. As far as the contamination detection, we know that we're running up against potential misdiagnosis or clear misdiagnosis in the case of a pure contamination issue. And of course, with sample mismatch or sample swaps, certainly PGTA result accuracy can be affected. And so bringing embryo QC into the test really will improve the accuracy of PGTA. It will also, as a consequence, improve mosaicism detection. NGS labs, or labs that run PGTA based on NGS technology, have in the past encountered criticism that mosaicism is overcalled, that we overdiagnose mosaicism. And this embryo QC, the ability to identify external contamination, will really help us address this problem, because imagine we have an aneuploid sample, an aneuploid embryo, and it's got some amount of contamination, and that will be euploid. The contamination will be euploid. That's going to show up as a mosaic, perhaps a high level or a low level, depending on how much external DNA contamination there is. And so when we can identify and call out a contamination issue, you can be sure that the mosaicism that's reported on the report. So when you've ordered PGTA Plus, you can be very confident that the mosaicism that we have reported on your report is not an artifact coming from contamination. This was touched on briefly, I believe, by Julian at the beginning, but if you're a client of iGenomics, you will have access to our iGenomic Clinic dashboard. And this is where you can view and monitor all of your PGTA data in real time. This QC data is coming to our clinic dashboard very soon. And so that means that as you're running PGTA Plus, you'll be able to log on to your clinic dashboard and see your QC data. You'll be able to see your contamination data. You'll be able to see your sibling relatedness data. And obviously, great labs, great results. You can use these KPIs to your advantage, and you can use your QC data to track your KPIs and use it on your lab or in your lab. And of course, you can also use this data for inspections. You can take this data and use it and empower yourself in your lab. You can use it to show inspectors for accreditation, for CAP and CLIA, and you can really leverage the fact that you're doing a great job, that your embryo biopsies are clean, and that you're not performing sample mix-ups. And another interesting point that's come up is that As you're well aware, any clinical procedure that you perform requires a clinical reason. And so when you're holding a report in your hand that has QC information and is clearly providing you with a reason to rebiopsy a sample, this is important and there's value in this as well. You can see that here. We've just given you an example of a PGTA plus report. And you can see here that sample 1 reported as euploid, sample 2 was triploid XXY, sample 3 was an aneuploid, a high mosaic, monosomy 6, and sample 4 was euploid. But look at the embryo QC. Pass, 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 pass. This really empowers you and gives you that confirmation that these results are accurate. They're quite accurate as we can get them at this point. And that this, especially, that this high mosaic monosomy 6 is actually a true high mosaic monosomy that we're finding in that sample, and it's not contaminated with any exogenous DNA. I just want to quickly draw your attention here to this triploid sample. You'll notice that the pass has an asterisk beside it, and this will be true of any embryos that we find with SMART PGTA Plus that have a ploidy issue, so the haploids or the triploids. We can identify and analyze contamination, and in this case, we discarded the possibility of contamination because it passed, but we can't use triploid or haploid embryos in our sibling relatedness analysis. So that's the reason that pass has an asterisk beside it. I'm sure you can imagine there are many, many different examples of reports we could show you, and I'm sure you probably have lots of questions about what they might look like, and your scientific advisors are ready and waiting to take your questions, and so please do follow up with them, and they'd be happy to share sample reports and explain all of this to you in more depth. But I'm sure that another question that's weighing on your mind right now, and certainly when we go out and we visit and talk with clinics and with IVF lab teams and embryologists, everyone is feeling kind of anxious, and we acknowledge that contamination, the word itself, can be a little triggering. So how will iGenomX manage cases that have embryo QC issues? And again, just remembering that 0.8% coming from contamination and an estimated 0.2% coming from sample swaps or sample mix-ups. And so overall, we're expecting the incidence of embryo QC issues to be quite rare, around 1%. But how will we manage these cases? So if we have a case or a patient and a sample that we detect contamination, the first thing we do is a full review on the iGenomX side. We look at the run, we look at other samples in the run. Once this sample has passed our internal review, we will generate and release the report to the clinic. Immediately following that, someone from the scientific team will get in touch with you to discuss it further. The reason that we generate and release the report and then follow up with the scientific team is because we believe that we have at least some embryonic DNA in that tube. We've detected 2 genomes. And so how will you manage these types of results clinically? Again, something that your scientific advisor would be happy to talk to you in more depth about, and please drop questions that you have in the chat. Our reasoning here is that euploid embryos should always be rebiopsied, of course, because euploid embryos are the ones we're going to transfer. What if that we've identified 2 genomes, but only 10% is embryo and 90% is maternal cell? That euploid result might make us a bit nervous. So any euploid result that has contamination, we do recommend that it be rebiopsied. Aneuploid embryos may or may not need to be rebiopsied depending on sort of your clinic preference and your clinic protocol, because again, remember that we're assuming that any external DNA is euploid, and if the embryo itself is still resulting as aneuploid, it may be less likely that you want to rebiopsy. A caveat to that might be that you might want to determine if it is in fact a low-level mosaic, or is it a high-level mosaic that would be low-level if not for the contamination. So there are some nuances there, but in general, just keeping it simple, this is the recommendations that we're making and suggesting at this time. And what about the sibling mismatch question? If we find a mismatch in a patient's cohort of embryos, again, we always start with our internal review at iGenomX. But unlike contamination, we do not issue or generate a report. we get in touch with you right away. We get in touch with the clinic, the lab director, the embryology team, and we give you a heads up about what we've found, and we try to help troubleshoot and figure out the situation. If we can discover some issue where this mismatch came from, once everything's been straightened out, we can issue the report upon request with the issue corrected. The rationale for us here in not releasing a report is because the result we obtained doesn't represent the tested embryo. It was completely contaminated, or it was completely mixed up. So we do recommend that any embryos that were identified as a sample mix-up should be rebiopsied regardless of what the result is, because we really can't trust on the result that we received. Okay, so I'm going to turn the floor back over to Alyssa Snyder. And I think, Alyssa, you want me to advance the slides?
Yeah, I'll just keep using your slides. That's fine. Thanks so much. And we've talked quite a bit about how our scientific team can help troubleshoot these issues of contamination and sibling mismatch. So the clinician should feel that they are fully supported by iGenomX in working through this PGTA plus additional information that you'll be providing. And I also wanted to underscore and share with you that our genetic counseling team is also here, both to speak with clinics and to speak with patients as well. Go ahead. So as always, but especially we're talking about PGTA plus, our genetic counseling team is here to support you and your patients. This is our iGenomX genetic counseling page. Alyssa, if you could click one more, we've got a little animation here. If you're quick on the click with your phone, we've got a QR code here that you can scan, and this will take you to our genetic counseling webpage. And you'll see several different options here where you can register for a pre-test PGTA webinar. You can schedule a post-test results appointment, and then our contact information is there. And at the bottom, we have some educational materials or results guides. They're PDFs. So this is on our website, or you can access it with a QR code here. Go ahead. We have pre-test counseling webinars available that incorporates information about PGTA+. These are live webinars that can accommodate up to 100 patients. This allows us to reach patients in remote locations and also provide the most up-to-date and current information because they are live and continually updated. They remain continually anonymous, and they have the advantage of having a Q&A session at the end, so patients have the opportunity to ask their questions live. We can track attendance, and at the conclusion of the webinar, provide consultation notes to the clinic saying, dear physician, your patient attended this webinar on this date, and that documentation can be placed in the patient's file. This is a webinar designed for patients, but you are welcome to register yourself as well, so you can get the full patient experience. You can see when the next available webinar might be for your patient, and you can even use it to educate new staff on PGTA. So a little QR code there is present as well. I have an animation if you wouldn't mind, Alyssa. At the conclusion of the webinar, a patient will receive a handout summarizing the information that was discussed, and so they will be able to access this information later on in case they forget. Go ahead. And we can also review results with patients, and they can go to the same website, click on schedule a results appointment to automatically schedule with one of our genetic counselors. Go ahead. There are two guides that we can provide. One is a general results guide, which will incorporate SMART PGTA as well as SMART PGTA Plus, and then its sister results guide specifically for Mosaic results. There's that QR code there as well. And thank you so much, everyone, for attending. That's the conclusion of our webinar, but we'll be happy to stay on for Q&A so we can answer all your questions.
And Alyssa, I'm very surprised with the number of questions that we have. It's been a lot. It's very nice. I had a chance to reply to some that are easy to reply. Regarding the task requisition form, or what is the difference from SMART PGTA to the regular PGTA. So those questions were easy to answer. So I just did. We do have others, Shannon, that we need to continue. And again, I want to appreciate the consistency of our guests. So our number stays the same. They came through all the presentation the entire time. It was amazing. So Shannon, we need you here. We do have questions regarding risks of contaminations from conventional IVF versus ICSI. So this is one of the first questions that we received. So I really want to hear your comment about that. And other thing that we have here is labeling the tubes after biopsy versus siblings mismatch. Like the procedure inside the laboratory, if you have a problem of labeling embryo number 4 versus embryo number 3, but they are actually the embryos from the same cohort, right? So we are not going to identify that. There is still a problem in the moment of the report, but we are not able to identify because they are still siblings. So add your comments to the procedure inside of the laboratory to help us.
Okay, I guess question number 1, there is risk of contamination with both conventional IVF and ICSI. I don't know that one is necessarily that much more prone to it than another. You would assume that conventional IVF would be more prone to having maternal cells remain, but some get very, very clean depending on the lab you work in. And some people don't mind leaving cells on when they're stripping for ICSI. So it can go both ways. So I think the idea is that we do want to minimize any maternal cells, any cumulus cells remaining on the eggs if you have a PGT case, regardless of your type of insemination used. So just make sure to clean those eggs very, very well, whether it's during stripping for ICSI or post-insemination prior to FERT check for conventional IVF. So make sure you get them as clean as possible if that's going to be a PGTA case. So on to the second question, the procedural question. Every lab needs to develop a very robust organization for labeling their tubes. Label your tubes first, stay organized, don't put them randomly in your rack and just have unlabeled tubes and just pick up a tube, tube it, and then label it and throw it back in the rack in whichever spot it happens to go to. Be organized, have a spot where you have your untubed tubes. Does that make sense? and then move them to another spot where once you have cells tubed, have them in another spot. That way you can easily see how they're organized. I personally prefer to label tubes prior to tubing, so they're lined up and in order, then tube all my cells, then they're all lined up afterwards. I heard from one lab recently, what they like to do is take a picture once they're complete, how they load their tubes into the rack that they're going to send off to iGenomics, take a picture so they can see 1, 2, 3, 4, etc., and add that into their patient's chart. So that gives them that final kind of check off of these are how this patient's tubes were organized. And I thought that was a very, very good thing that other labs should probably pick up on.
It's great. There is one thing that I believe that we should mention as well, Shannon, regarding all the questions that we are receiving here. We are not bringing this to the market just to create more confusion, or actually the intention is the other way around, is to have a more accurate test, but also with all your experience and with the work together between Vitrolife Group and iGenomics, do you want to mention the possibilities that we have for training, for troubleshooting?
Yes. Yes. So I guess
the workshops that we have. So if you want to just mention.
Yes. So we have introduced now, since we came together as Vitrolife Group and have both Vitrolife and iGenomics, we have launched a series of biopsy workshops, and we've done 3 so far over the past year and a half, and these will continue into next year and beyond. And they're called Biopsy 2.0. So they are biopsy workshops that are targeting not the super beginner necessarily, not the novice just learning biopsy, but someone who has started biopsy and needs to improve just the tools in their toolbox, learn other techniques. We all learn from one person in our lab and what's their favorite technique, but there are other techniques out there. So this is a workshop that teaches different techniques, gets you a little bit more in-depth information about what happens within the genetics lab, kind of behind the scenes, so you can understand what goes on there and how what we do in the laboratory might influence those results that we get. And so you have 2 days of both lecture and hands-on, and that brings both the Vitrolife team and the iGenomX team together. We also have the ability to go out to a lab that might just need some specific training. Maybe they have seen a spike in their no-read rate, and we can go help them troubleshoot to figure that out. Anything like that. So kind of a custom-made workshop just for your laboratory, if that's what you need. So all of these things can happen. Whatever support you might need that is specific to your laboratory, we can build that for you and bring that knowledge to you between the scientific team on either the iGenomX side or the Vitrolife side.
Yes, so it's important for everybody know that we are very concerned and working together to have the best results as possible. I do have another questions here that are very interesting. I'm looking to the time. It's 4 minutes past 5. Perhaps we can answer just 1 more or 2 questions. Alyssa, we have here comments that perhaps you, Alyssa Snyder, you should create another webinar or training to explain to us your discrepancy protocol to investigate. It's going to be interesting for all the ones doing PGTA and understand how we are able to identify the reasons for discrepancy. So we do have a suggestion for a new webinar here. We also have some questions regarding if PGTA Plus is more expensive than regular PGTA, and I'm recommending you to reach out to your sales rep, and definitely you can see difference in prices, but then you need to understand how it applies to your patients. Other questions that we have here is regarding the tests. So they are both coming in the same task requisition. You can test and select the one that you prefer. Both of them, it's going to be in the same task requisition, but you can select it. We do have the question about NGS and SNP being run in the same platform, or if they are 2 separate tests. So I just want to reinforce that we are running 2 separate tests. They are 2 independent tests that it's providing us the different sources of the same, about the same embryo, like 2 sources of results from the same DNA. There is pros and cons for this technology. This way you don't have the entire test dependent on a NGS run. So we don't have all the 2 tests dependent on the same run. So they can prevent us to failure in one and the other. But by the other hand, of course, we are running in separate, so we have the double the work to get the results. This is a technical operations decision that we made internally. But both of the results, all the data, it's combined in the same algorithm, and in the moments that we are analyzing the embryo result, the 2 source of the results, they are combining in the same reporting system. So this is also important to say. There is a question here regarding collecting saliva and collecting DNA from parental DNA for detecting any other type of a not relatedness, that it's not necessarily only with the siblings. Any of you, again, Shannon, perhaps with the quality inside of the laboratory, or any of you wants to comment as well, the parental origin of the embryos. So if I can say this right.
I have some thoughts.
Okay.
Regarding the parent of origin of aneuploidy, I know that some people are very interested to know this, with the intention being that there's clinical utility in being able to choose an egg or a sperm donor. And I would argue that a better analysis is just to look at the sperm directly. So if you're thinking that I'm going to choose to use a sperm donor because of a higher incidence of aneuploidy coming from sperm, let's think about it. Let's say you had 10 embryos, and we would expect that 90% of the aneuploidies usually come from the egg. So we would expect that 9 of those embryos would have maternal aneuploidies if they were all aneuploid anyway. If you see 50-50, you would say that's higher than expected for aneuploidies to come from the sperm. Is that really enough to make that decision about using a sperm donor? I would argue not. And most likely, if you have 10 embryos, only a few of them are going to be aneuploid. And so you're really not going to be able to get the statistical power you need to be able to use that information to make such a big decision about sperm donation. So there's another analysis that iGenomics can offer called sperm aneuploidy testing, which is a direct analysis of the sperm. And we would offer that as a more direct way of assessing the sperm if that's the question of being able to use a sperm donor.
Lisa, the person who asked me the question first just rewrote it and say more as a final check for gamete mix up.
Gotcha. So in order to be able to detect gamete mix-up, we would have to be able to get DNA from the egg source and the sperm source, so patient and partner, which we decided when we were building our platform that would put undue pressure on our clients and undue pressure on ourselves as a routine clinical test. So we made the decision that we would not make that part of our basic platform. If we're thinking about, everything in the media, most of what we see is actually wrong embryo transfer. It's very seldom that the embryos don't belong to patient or partner because of the incorrect selection of the eggs or the sperm. That being said, guys, iGenomics is here to support you. If you have a question or concern about what gamete source was used, we have other ways of helping than a clinical test. So we can run, for example, STR analysis on it. So please let us know if you have concerns, and we can certainly help confirm the egg and the sperm source.
We still have a consistent number of guests here. So I think that I'm going with the last question. And then I'm taking note with all the other ones that we were not able to answer. So we are going to send an email and reply to everyone. But the other question, it's about the 2.1 PN and the 3 PN. So a question is related to, do you see difference between 2.1 and 3 PN? Because we show our numbers in the poster, like combining both of them with the risk, the chance of a euploid. I don't know, Shannon, what is the frequency of 2.1 PNs in the lab? Alyssa, you presented the numbers, if you want to comment that as well.
Do you want to answer,
first.
Shannon?
I guess I'm first. That tends to be very patient specific. It's not huge where you see the 2.1 PN. You're more likely to see 3 PN. It does happen, of course, but 3 PNs are probably more common than the 2.1.
And the data that I presented didn't include the breakdown between 2.1s and 3 PNs in terms of percentages, so we would have to ask the authors directly, which we could do.
So as I said, I think that we should close our session now. I tried to reply the ones that I could. There were some questions also here regarding the percentage of mosaicism in both platforms. Oh, okay, this is the last one. Is the questions coming? Have you seen different percentage of mosaicism in both platforms, comparing PGTA with the PGTA plus? What is your guess? Magwood, Snyder? We're starting plus now, so we still don't have data for that, but what is the guess?
Yeah, I mean, I'll jump in and Alyssa Snyder can back me up or add to it. But yeah, we do expect to see a lower incidence of mosaicism, because if we are removing I don't want to call them false mosaics, but procedural mosaics, let's call them that. Mosaics that are resulting from a contamination issue. So if we take those out of the equation, we would expect to see the incidence of mosaicism with PGTA plus to be slightly lower. And I think that's a more biological representation of the real mosaicism that exists.
Great.
Nothing to add?
That's
Nothing
good.
else? Okay, so thank you so much. Thank you for participating. You that have been here with us, it was a pleasure. You're going to receive a copy of the recording. Don't be shy, share with your colleagues. See you next time.
Thank you.
Thanks, everyone.